Muy buenas tardes para todos. El Jardín Botánico de Bogotá, José Celestino Mutis, y en nombre de las directivas de nuestra institución, en cabeza de la doctora Marta Liliana Perdomo y la subdirectora científica Claudia Alexandra Pinzón, le damos una cordial bienvenida en el marco del mes de la relación planta-animal. Daremos hoy entonces una conferencia que se ha titulado Ex-Situ Species Conservation, Predicting Plant Survival in Botanical Gardens, que tiene por objetivo presentar experiencias de investigación en relación con la adaptación de especies en colecciones vivas. Este evento tendrá una duración máxima de 2 horas y durante el desarrollo de este evento los estaremos acompañando María Eugenia Torres, María Claudia Pacheco, Nancy Mejía, Iván Jiménez y quien les habla Magda Bermúdez. Asimismo, les informamos que en el chat de preguntas estaremos enviando un link para que ustedes se registren su asistencia. También los invitamos a que nos registren o registren sus dudas en este mismo sitio y preferiblemente y hagan sus preguntas o escriban sus preguntas en inglés. Cabe aclarar que este evento será dado por Georgia Thomas y será en inglés. En ese sentido, entonces, me permito presentar a Georgia Thomas. Ella es bióloga y tiene una maestría en botánica de la Universidad Wyoming. Actualmente trabaja para la División de Registro de Plantas del Departamento de Horticultura del Jardín Botánico de Missouri. Su trabajo se centra en la conservación ex-situ de especies atípicas y amenazadas en todo el mundo. Le damos una bienvenida especial a ella. Georgia, welcome.
Thank you for having me today. Are you able to see my slides now?
Yes.
Hey,
Very good.
Thank you again for the opportunity to share the results of my work. Thanks again for having me. I apologize I'm unable to give this talk in Spanish. So the talk today will be titled Ex-Situ Species Conservation, Predicting Plant Survival in Botanical Gardens. And my collaborators on this project include Andrew Wyatt, who is the Vice President of Horticulture, and Rebecca Suker, who is the Living Collections Manager at the Missouri Botanical Garden. My other collaborator is with us today. His name is Iván Jiménez, and he also works for the Missouri Botanical Garden and their Center for Conservation, Sustainability, and Development as a research scientist. The talk today will be broken down into 5. I'll start with an introduction to ex-situ conservation, then describe our study objectives. We are interested in predicting the survival of plants outdoors at botanical gardens. Then I will go into our methods and introduce you to the components of our mini models. In the results section, I will present to you our findings showing predicted survival in gardens, both in terms of survival curves and prediction maps. Finally, I will conclude the talk with a summary of our findings and its implications for the broader botanical garden community. In our study, which I'll summarize today, we were interested in exploring the use of outdoor landscapes in botanical gardens for ex-situ conservation. Ex-situ conservation may be defined as protecting species outside their natural habitat. It plays an important role as a safety backup against extinction in the wild. This is reflected in Target 8 of the GSPC, which is the Global Strategy for Plant Conservation. This strategy calls for at least 75% of threatened plants species to be conserved in ex-situ collections. There are multiple forms of ex-situ collections. The most common ones are seed banks and botanical gardens. Seed banks obviously are known for preserving seeds, while botanical gardens preserve whole growing plants. I will refer to botanical gardens in a larger sense to include arboreta and any other documented ex-situ collection of whole-growing plants. Botanical gardens can be particularly important in ex-situ conservation of plants which produce exceptional seeds. Exceptional species, those that produce seeds that cannot survive standard seed banking procedures such as drying and freezing. Species which produce such seeds, including those that are recalcitrant, which are those that don't survive the drying process, represent approximately 10% of all angiosperms and 36% of critically endangered plants. Botanical gardens can play an important role in ex-situ conservation of these species. Here I will go into more detail on the function of living collections of botanical gardens, the functions living botanical gardens play in ex-situ conservation. First, botanical gardens hold species diversity. We can think of these botanical gardens as a living library, where not all plants are used at the same time, but they are held and readily accessible when needed. Gardens target and build collections to conserve genetic diversity, as well as species diversity. This is particularly important for preserving species which are threatened in the wild and those with exceptional seeds. Living collections can prevent a species from going extinct or provide stock for reintroduction projects. Other benefits of living collections and ex-situ conservation include include the knowledge we gain from bringing plants into a cultivated environment. Bringing a plant into a cultivated environment provides a lot of opportunity to learn about species. Seed cleaning protocols, seed storage protocols, viability testing, propagation protocols, cultivation protocols can all be developed when a seed is in a cultivated environment. It's a great opportunity to learn more about the species. Furthermore, having living collections congregated in one location allows researchers to sample a wide range of highly documented species in one space, saving, of course, time and money. Finally, living collections play a role in educating the public. Displaying rare plants boosts understanding of the importance of diversity and conservation, and provides for discussion around habitat conservation and conservation projects in general. The Missouri Botanical Garden has been very active in this type of ex-situ conservation. For some rare and threatened focal species, we have worked to preserve all known genetic variation of the species. This is critical for keeping species from going extinct. On this slide here is an example of a critically endangered tree species from Africa, Chromia gigas. It's grown at indoor facilities at the garden. It is only known from 19 individuals in the wild. We're able to track all our propules down to the mother plant and truly understand this this species. This is a very intensive process, and it's important to keep in mind that indoor facilities where this particular species reside are expensive to maintain. Therefore, in our study, I'll summarize today, we're interested in exploring the use of outdoor landscapes in botanical gardens for conservation. Although often conceived of as ornamental and educational displays, outdoor landscapes hold a large proportion of the plants in botanical gardens. For example, more than 32,000 individual outdoor plantings are inventoried at MBG. Outdoor landscapes represent some untapped potential for ex-situ conservation. It's a large number of plants outdoors. Next, I'll go into the specific objectives of our study. The impetus of our research was to explore the suitability of these outdoor landscapes at botanical gardens for ex-situ conservation. We modeled plant survival based on differences between climate in situ and in situ, that's in the wild, versus ex-situ in botanical gardens. Finally, we wanted to use models to predict propagation, propagule survival outdoors at botanical gardens. This result would make for better informed horticulture decisions and help us better plan our collections. Traditionally, horticulturists have used hardiness zones to rate the suitability of a climate for different species. The Missouri Botanical Garden falls between zone 6 and 7. Missouri in St. Louis, where the Botanical Garden is located in St. Louis, has a highly continental climate with really cold winters and really hot summers. We experience extremes in St. Louis. Hardiness zones utilize average extreme minimum temperature, as I've circled here. Regions are given hardiness zones based on this extreme minimum temperature. However, there's no standardized method for assessing a plant's ability to just survive and thrive in a particular hardiness zone. Qualitative observations are made of these individual species and reported. A grower will simply report that a individual rose, for example, can survive down to a hardiness zone 2. For our study, we wanted to assess this problem in a more quantitative approach using data gathered from the garden. We hope to be able to predict a species survival at the botanical garden. A plant simply being able to live for a given period of time is the minimum needed for ex-situ conservation. We need to know we can keep these plants alive before we invest in ex-situ conservation. Next, I will discuss our study's methods, starting with our response variable, survival, then move into our models and the predictive variables. I will start by describing the response variable, survival. Our study relied heavily on data from the Living Collections Management System. This is the horticulture database at MBJ. It is essentially a system that tracks all plants growing in the garden, including information on the source of each plant, its propagation protocols, and the plant's taxonomy. Out of all the data in the LCMS, which is the Living Collections Management System, for our study, we were interested in the number of days individual plants survived outdoors in displays at the Missouri Botanical Garden. This is what's illustrated in this particular slide. The number of days a plant survived was how we measured the plant's response to climatic conditions, aka the success of the plant in the particular climate of the garden. Survival is illustrated on this graphic as on the in this graphic. On the horizontal axis is time, and a series of individual plants are shown on the y-axis. The black dots show the date for which the plant was planted outdoors, and then the open circular symbols show the time of death. In this illustration, individual plants 1 and 4 are still alive today. This is because this is an ongoing and growing data set. All study plants were grown in displays on the garden grounds where the public can view them. They were not specifically grown for this study. So here on this this orange bracket, it shows the variability we modeled in the study, which is survival time outdoors. But we were not interested in survival of just any plant. We focused on plants grown outdoors from seeds collected from propagules collected in the wild. This is to mimic ex-situ conservation. So wild collected plants. We focused on survival data from 1,184 plants grown outdoors at MBG. This represented 410 species sourced from 530 localities around the world. These localities are shown on the map as orange dots. As you see, most of the propagules were collected in temperate regions in the northern hemisphere. It's important to note that the study specimens only account for a small number of the plants we have in the garden. We have more than 1,000 plants outdoors at Missouri Botanical Garden. For this study, we were limited to the sessions to which we knew the coordinates where they were collected. This really highlights the importance of record keeping in botanical gardens and the preservation of provenance information. We would not have been able to do this study without this kind of data. Next, I will describe the 3 broad categories of models we used. We modeled survival probability at the garden using more than 300 different models. These 300 models can be broadly classified into 3 kinds. The first kind of models we call locality models. That's shown at the top of the slide. This is shown at the top. This model looked at the survival probability of a given plant as a function of the climate at the particular location where the respective propagules were collected. These models were designed to simulate local adaptation. The second 2 broad categories of models can be termed species models, and they're shown at the bottom of the slide. These models regard survival probability as a function of the climatic distribution of a species. Both of these species models conceptualize suitability to the garden's climate as a functional trait of the species. Both of these models took notes from the theory behind species distribution modeling, SDMs. So the second model looked at survival as a function of the central tendency of the climate, the median climate for the distribution of the species. While the third model imagines survival probability as a proportion of the climatic distribution of a species. So this is a central tendency model and a proportional model. So in total, we have those locality models, the species central tendency model, and the species proportion models. In the next few slides, I'll go into these in more detail. So I would like to illustrate the features of these models using a sample species. And we're going to today talk about Engelmann's milkweed, Asclepias engelmannii. This plant is grown at MBG in the rock garden as indicated by the black diamond. The seeds to grow the plant were sourced from the locality indicated by this square, the dark red square. And finally, the native geographic range of Asclepias engelmannii is indicated by the round symbols, and there's a convex polygon that encloses them. Each round symbol represents a known occurrence of Engelmann's milkweed, Asclepias engelmannii. We source these known occurrences from GBIF. So here's this first broad kind of survival model. That's the locality model. These models calculate the difference between climate. These models calculate the difference in climate between the propagule collection locality and that of MBG. And then these differences were used to predict plant survival. The other two broad categories of models, the species median model and species proportion model, are shown here. These two types of species models calculated the difference between MBG and the whole native geographic distribution of the species, as conceptualized as a median and proportion respectively. In this slide, I just showed one climatic variable, annual mean temperature. But we included other models, other climatic variables in our study, and I will go into that next. So I will just start describing the predictive variables used in the study, the climatic variables. I will describe them in light of these three general models I just described. As we examine climatic differences, we wanted to consider the effects of horticultural care on the plants and outdoor landscapes. We made the assumption that the care by our gardeners allows plants to grow that would not be able to in nature. Horticulture care includes activities such as irrigation, soil augmentation to create beds with better drainage, and plant maintenance such as weeding, which eliminates competition. To capture these conditions, we developed what I will call horticulturally informed climatic difference variables. These I'll go over in the next few slides. I will start with looking at how we measure climatic difference variables for our precipitation variables. We made the assumption that water could be added through irrigation at the garden, but not taken away. If a plant lives in a really wet region, such as a rainforest, our horticulture staff can mimic those conditions through irrigation. However, it is much harder to create conditions of low levels of precipitation, such as that of the climate in deserts. Therefore, using these assumptions, we created these one-sided difference variables. The equation above represents the difference in annual mean precipitation at MBG, which is 980 millimeters annually, and that where the plants occur. If the annual mean precipitation is greater than St. Louis, the difference, we know the difference as 0. While if the precipitation levels at the occurrence site were less than that of St. Louis, we found the difference. This is illustrated in the map to the left. We represented the climatic difference, excuse me. See how these wet areas are represented in green with the climatic difference is 0, even though areas such as the Amazon get a lot more rain than St. Louis. But the differences do increase in the other direction, where the pink areas get a lot less precipitation, annual precipitation, than St. Louis or the Missouri Botanical Garden. Now, if we're going back to our previous slides, we just discussed having 3 general types of models. Those models were based on 3 general types of models. Those models based on the collection locality and the distribution of the species. Well, these models are represented in the figure on the left for our sample species, Asclepias engelmannii. In this figure, annual precipitation is listed on the x-axis, while the density of known occurrences is shown on the y-axis. The density of occurrences at a specific precipitation level is shown in this gray curve. You can see a high number of Asclepias engelmannii occurrences, around 500 millimeters of precipitation a year. The lines in this figure represent precipitation at points of interest. The yellow dashed line represents the precipitation levels at MBG. which again, as I talked about earlier, is around 980 millimeters annually. So next, I'm going to talk about the models based on propule collection location. That's illustrated right here, with the arrow. So in this figure, the red line represents the precipitation at the propule collection locality, which is that red line. And the difference between the two is the delta annual mean precipitation. So the difference between MBG, which is this yellow dashed line, and the precipitation at the propule collection locality, which is this red line, is our difference variable. So now let's move to the two species distribution based model. So we've talked now about the locality based model. Let's talk now about this species central tendency model and species proportion models. Remember that the density of known occurrences is represented in this gray slide, and this dash, this kind of dotted red dotted line represents the median level of precipitation of the density of the occurrences. So we calculate the difference between the species median and MBG using that same equation above. Finally, we also calculated proportional models, so we calculated the proportion of species that had precipitation less than MBG. This is that gray shaded polygon. Now the assumptions for calculating horticulturally informed climatic difference variables for precipitation were relatively obvious based on our management capabilities. But we also wanted to account for horticulture care and create one-sided difference variables for temperature. We hypothesized that the extreme high and colds of MBG were limiting factors. Anything more extreme than MBG was not limiting. Plants in these conditions benefited from horticulture care. So seen here, the minimum temperature of the coldest month at MBG is negative 6.4 degrees Celsius, so well below freezing. If the minimum temperature of the coldest month was colder than MBG, the climatic difference was 0. You can see this in the green section of the map and the white section of the polygon. This is why the sections of the Arctic that are green with a difference of 0, although they experience much colder minimum temperatures of the coldest month than St. Louis. Similarly, we calculated horticulturally informed climatic differences for a maximum temperature of the warmest month, but in the opposite direction, as MBG experiences hot summers with a high of 32 degrees Celsius. We did not believe temperatures more extreme than St. Louis would be limiting. We thought we could manage this through horticulture care. However, plants from places which do not experience such extremes in hot temperatures could suffer in the heat. In addition to these variables, we also utilize climatic difference variables to create horticulturally informed climatic difference variables for other climate measurements such as mean annual temperature, precipitation of the driest month, and precipitation of the wettest month. So in total, we tested 378 mixed effects Cox models. Remember, we are modeling survival outdoors at the Botanical Garden. To create our 378 models, we took our 3 general approaches to measuring climatic differences. Again, the 3 approaches were the locality-based model, the species median model, and the species proportion models. For each of these three approaches, we utilized 63 combinations of our climatic difference variables. For example, some of the combinations of climatic difference variables are shown in this slide. We used every possible permutation. That's how we got up to 63. We also needed to account for the fact that not all species are created equal. Some species of plants have inherently longer lifespans than other species of plants. We accounted for this in two ways by using random effects. In one set of models, we assumed lifespan length is an evolutionarily conserved trait. In this set of models, we accounted for phylogenetic relatedness as a random effect. And the other set of models, we calculated the random effect at the species level. We did not assume that lifespan is evolutionarily related in that set of models. Finally, there's one more variable which I have not yet discussed, but we include in every single one of these 378 models. This variable accounted for the difference in lifespan between woody and non-woody species. Generally, woody species are known to live longer than non-woody species, and they have longer generation times. Therefore, each species in our study was coded as either woody or non-woody. For example, Asclepias engelmannii is a non-woody plant. All right, now for the exciting stuff. I'll start describing the results of our study. I'll start by examining our best models. So to find our best models, find the models with the most empirical support, we compared AIC values. Delta AIC is shown on the y-axis of the top graph. Each of the models is represented on the x-axis or with a distinct symbol. The bottom panel on this slide shows the climatic difference variables represented in each of the models. So to interpret delta AIC, the smaller the delta AIC, the greater the empirical support. These are the models at the top of the panel, the top of the top panel. Generally, there's a rule of thumb that states that models with a delta AIC of less than 7 have the same amount of empirical support. Therefore, as you can see, our results show that models which use species as a random effect did much better than those which use phylogenetic relatedness as random effect. So therefore, this finding shows no phylogenetic signal within our study there's no phylogenetic signal or evolutionary conservation of plant survival length for species within our study. Here you can see that our top models use the propagule collection locality models. These are the models that have the most empirical support and are represented by the blue circles. This result highlights the importance of local populations and local adaptation in predicting plant survival. Now, based on these best set of models, the local models, what were the significant climatic difference variables in the models with the highest empirical support? So this bottom panel in the figure shows the horticulturally informed climatic difference variables on the left. The dark black dots show the variables that occur in the models with the highest empirical support. You can see minimum temperature of the coldest month occurred in most models with high levels of empirical support. What is not shown in this figure is that the difference in minimum temperature of the coldest month, best variable, also had a similar weight and significance on survival in all of the best models. Another common variable was maximum temperature of the warmest month. It too had a significant impact on survival. As for precipitation, a precipitation variable was included in most of the best models, but it may or may not have been significant. In the following set of slides, I'm going to present the results from one of our best models. This is going to be model 9. It's circled here in yellow. This model included the following climatic difference variables. Minimum temperature the coldest month, maximum temperature the warmest month, precipitation the driest month, and also annual mean temperature. However, annual mean temperature was not significant in these models. So using that model 9, I will show the results in terms of the response variable survival. I'll start by showing survival curves. So let's go over how survival curves work. In these graphs, the number of days is on the x-axis. On the y-axis is the probability a plant is alive. So each of these different curves represents different climatic scenarios. The top dark black line represents survival of non-woody species which come from climates the same as that of St. Louis. You can see even for plants that come from the same climate as the Missouri Botanical Garden, MBG, there is a loss in survival over time. At around 4,000 days, the probability of survival is around 80%. In this figure, we're examining the effect of differences in minimum temperature of the coldest month on survival. So if all other climate variables are held the same as experienced at the Botanical Garden, we can see the impact on increasing the difference in minimum temperature of the coldest month, increasing the difference between the collection locality and the Missouri Botanical Garden. For example, in the lightest gray climate scenario, which is the lowest curve, we are predicting survival of plants from tropicals from sites that were almost 20 degrees Celsius warmer than Missouri Botanical Garden. As you can see, most of these plantings are dead after 2,000 days. There's a low level of survival. Now, using the same climate scenarios, let's examine the difference between survival in woody and non-woody plants. Woody plants tend to live much longer than non-woody plants. As comparing the left graphic to the right graphic. This has a potential to affect ex-situ conservation decisions. Woody plants may be easier to grow in the short term. However, keep in mind that woody plants take longer to reach maturity and survival will decrease over time. Here we look at the difference in climatic variables between maximum temperature the warmest month. As you can see, propagules collected from locations where the warmest month is colder than MBG also do not have a high probability of survival. However, differences in maximum temperature the warmest month do not have quite as strong an effect on survival as the difference in minimum temperature the coldest month. Finally, these are the effects of precipitation. As the difference in precipitation increases, the survival probability decreases. However, within the range of the values studied, this effect is not as strong. However, climate variables do not operate independently of one another. Temperature does not change while precipitation levels stay the same. So I will present some maps that predict the survival of plants collected from specific localities. I will start with our example species, Asclepias engelmannii. So here are the results. The colors in the map on the left-hand side of the slide represent survival probabilities, according to the scale on the right-hand side of the slide. Each color on the map corresponds to a particular survival curve, also shown on this figure. So on the map, red means a low probability of survival through time and blue a high probability of survival through time. As you can see, survival probability varies extensively within the range of Engelmann's milkweed. Again, this emphasizes the importance of collecting from a site of the collection site for your particular botanical garden. So in our particular site where we collected, our propagules were collected from, is represented by the red square, and it did fairly well in our garden and had relatively high levels of survival. So here I am presenting the results from the mean non-woody species in our study. This slide shows predicted survival for non-woody species. As you may see, predicted survival is fairly high for propagules collected from northern latitudes in eastern North America, western North America, Eastern Asia, parts of Western Asia, and the Mediterranean. And then I should point out that the gray areas on the map are those with climates beyond the range of our data, so we refrain from displaying predictions in those areas. So another important result, which we sort of already touched on, is the contrast between woody and non-woody species. This slide shows the predicted survival for woody species. And then although the spatial patterns are similar to that shown for non-woody species, you may see that predicted survival is much higher for the woody species than the non-woody species. This difference likely reflects the general tendency of woody plants to reproduce later in life and therefore live longer. All right, so what should we learn from our study? One of the main results from the study is that locality models are much better than species models. As I said, this result implies that climatic suitability of botanical gardens may be best thought of in terms of the propagule's particular collection locality, as opposed to the function of the species as a whole. Local adaptation plays an important role in survival outdoors at botanical gardens and has management implications. Another important result is that survival in outdoor landscapes at the gardens was fairly high for plants sourced from a wide range of northern latitudes. So plants sourced from these landscapes could contribute significantly to our ex-situ conservation efforts, with a decent chance of survival. So at MBG, Missouri Botanical Garden, we are using these results to help guide our ex-situ conservation efforts and target regions and individual plants for conservation focus. A third important result of our study is that the best models predicted higher survival for woody than non-woody plants. Thus, it would seem easier to maintain ex-situ populations of woody species in the garden. Of course, maintaining large trees in a botanical garden may require long-term planning and substantial investment. But again, it's important to remember that smaller, younger plants may add value, may add conservation value to a living collection, just as seeds represent seed banks also add value to a collection. All right, finally, I'd like to mention that we believe this approach may be widely applicable to botanical gardens as a tool to increase the representation, plan to help increase the representation of species of conservation concern in living collections. We'd be interested in expanding the study. We would be interested in gathering survival data from other botanical gardens and seeing how horticultural care assumptions change at different institutions, and also how the impact of these important climatic difference variables varies from institution to institution, as each institution has its own base climate. Finally, more generally, we hope that botanical gardens consider the need for ex-situ conservation as they plan their outdoor landscape. For this study, I'd like to thank all the horticulture staff at the Missouri Botanical Garden for both their diligence in caring for our plants and their diligent record keeping. I'd also like to thank the research staff at Missouri Botanical Garden for their input on this project. So I believe we're going to open up to questions next. Thank you so much for bearing with me, and thank you for allowing me to give this presentation in English.
Oh my God. Thank you, Georgia. Bueno, voy a dar paso entonces en este momento a María Claudia. Ella nos va a ayudar.
Sí, creo que el micrófono de
Perdón.
Marta.
Sí, thank you, Georgia. Estaba diciendo que damos paso entonces a las preguntas que se generen por parte de los asistentes. Habíamos recordado que estas preferiblemente, en la medida de las posibilidades que ustedes puedan, hacer las preguntas en inglés. Igual aquí también tenemos el apoyo de María Claudia y de Iván Jiménez para lo correspondiente. Entonces, bueno, iniciamos. María Claudia, te doy paso para que puedas entonces hacerle las preguntas a Georgia.
Okay. Georgia, we have a question from Sandy, and this person asks, what kind of adaptations have you recognized on plants, keeping in mind the climate changes?
So there's just this general thought that plants might migrate in terms of as a response to climate change. And I think botanical gardens are a good place to kind of predict and examine, because we can move plants within botanical gardens and move them to different climates ahead of climate change. That's a great place to do studies to answer the question of what adaptations are important.
Thank you very much. And let me see if there is any other questions. The people is thanking you for this presentation. And let me see if someone else asks anything here. Okay. We don't have any other questions at the moment. I don't know if perhaps Ivan has any question to make to Georgia.
Sure, Georgia. How you mentioned at some point as the value of Ex-Situ populations, that one important thing was knowledge through cultivation in your earlier slides. Could you expand a little bit on what you mean by that? And I don't know if you have any particular example that could illustrate that point of how you gain knowledge through cultivation, and particularly of species of conservation concern.
Sure, there's just by having a plant in a place you can study it, there's really boundless information you can learn. There is this, I know at Missouri, I cannot remember the name of the species right now, but it's a pretty well-known water lily species that is, I believe now it's grew in thermal hot springs in Africa and is now extinct in the wild, but available in Ex-Situ collections from Kew, and we also have it at Missouri. And I know that when it was growing in the wild, people really truly didn't understand how to propagate it and how to encourage it to keep growing, if in fact we had appropriate in situ a habitat for it. And by having it at the gardens, we're able to learn how to propagate these seeds. I believe they need it may be that the seeds need to be dry on dry land before they will propagate from seeds. So it was a really great learning opportunity.
Thank you very much. And we have another two questions here. One of them is, is there any concern about invasive species during the studies that you mentioned in the presentation?
Absolutely. We absolutely do have some concerns about invasive species. However, generally, we want to first handle the potential for anything to become invasive through really tracking any plants we have outside, making sure that they don't seed and expand their range or expand within the bed which they're growing in. However, I really think that the risk is worth the reward is worth more than the risk in terms of invasiveness, at least at the small scale of doing this at botanical gardens, for the fact that we can learn about species response to learn about species and their potential to become invasive. And we also, as climates change, plants will be moving, so it's kind of a way to understand this ahead of time. And in some cases, really, ex-situ conservation is the only way to keep species from going extinct.
Perfect, thank you very much. And the other question we have here is, do you think soil biodiversity have a major role on local adaptations or in ecological range of species?
Yeah, absolutely. Soil has, thank you for the question. Absolutely, soil has an impact. One of the assumptions we made in this study, which I didn't talk about today, is that our horticulture staff within the range of the botanical garden chose the beds with the best possible soil mix for this particular species. It would be interesting to be able to model this, but we already had over 300 models. Adding soil in is another challenge. But certainly something we consider at the Missouri Botanical Garden. And I know that, for example, the rock garden and one of the bulb beds, the soil is absolutely augmented and brought in from the outside to help expand the range of plants we can grow.
Okay, thank you. And the last question we have is there any risk by bringing species from similar climate?
Yeah, that's absolutely this invasive risk. One thing we do at Missouri Botanical Garden is perform a weed risk assessment prior to bringing any plants in. So we will not bring any plants that are known to be invasive. And like I said, I do think this is an opportunity to learn what plants are potentially invasive on a very small scale. So if a gardener notices that a plant is spreading within its particular bed, it is removed quickly, and notes are made about that. And hopefully we can eventually be able to make those notes more visible to the public and create sort of an adaptive management strategy. I think this is particularly important as, like I said, plants will be shifting ranges because of climate change. Thank you for your question.
Thank you very much for your answer, Georgia. I see there is no more questions here. So
could I have one more question?
Okay.
So Georgia, in your conclusions, you mentioned that the Missouri Botanical Garden or plants brought from a broad range of northern latitudes can survive pretty well at the Missouri Botanical Garden. But if I may be mistaken, and maybe you can show the slides for the predictions across the world, because I think we're seeing the slides. But it seemed to me that that was true also for southern latitudes. But I also think that the Missouri Botanical Garden doesn't really work bringing propagules from southern latitudes. Is there a reason for that?
It's something we're certainly, so one, yes, you're correct that you can see these yellow and in this slide, these yellow and lighter blue areas are areas where we predict decent amount of survival in species. One, I would say that while we're really expanding our ex-situ conservation efforts, so around the world. And we would absolutely be interested in bringing plants in if we think we can grow them. I do know, for example, that we have some representation of some rare and threatened bulb species from the more Mediterranean and moderate climates in South Africa.
I think the predicted survival is even higher for trees from those southern regions.
Yes, absolutely. That's shown on this slide. Yeah, it would be very interesting to try to bring in some trees from for say example, the Andes. So mid elevation in the Andes might be fascinating. And I would assume the reason it's not happening is funding and connections, but we can work on that.
Thank you very much. I don't know if, do you have any other questions, perhaps, Ivan, or?
No, I don't have any further questions, thanks.
Okay, thank you. So thank you very much, Georgia, for your presentation and for assisting to this event. We will go ahead and have Magda on the screen.
Yeah, thank you. Thank you for accepting this invitation. Very, very good. Bueno, entonces vamos a dar cierre a este evento, pues no sin antes agradecer a todos los que nos acompañaron el día de hoy, especialmente a Georgia Thomas, sobre todo también a Iván Jiménez, quien nos acompañó en toda esta transmisión. Al Jardín Botánico de Missouri, especialmente por esta alianza interinstitucional. Y finalmente, entonces, damos gracias también a nuestro apoyo técnico desde la Subdirección Científica con María Claudia Pacheco, Nancy Mejía, María Eugenia Torres, y los invitamos a seguir conectados con nuestra agenda académica este próximo 26 de mayo con la charla que está titulada ¿Por qué el COVID-19 es una invasión biológica? A todos ustedes los esperamos entonces este día y nos despedimos por el día de hoy. Muchísimas gracias.
Gracias.